Assalamu alaikum. I'm Dr. Sana Amjad, and today is our first lecture of second module, which is embryology. Okay. So let's begin with this topic. Okay. Now this is a very important module, embryology, and you may get 10 to 15 questions from this module. A little bit tough because you have to memorize each and everything. You have to memorize at which day this develop, at which day this develop. So it's a little bit tricky, but you have to remember these things. ज़रूरी नहीं है कि आप सब पूरा का पूरा मॉड्यूल आप बड़ा अच्छा सा याद करें और हर चीज़ याद नहीं रख सकते, लेकिन कुछ वेरी इंपॉर्टेंट पॉइंट्स हैं वो बिल्कुल स्किप नहीं करने हैं, ठीक है, वो बिल्कुल याद रखने हैं, टिप्स पे होने चाहिए, तो इनके नोट्स बनाएं जैसे भी आपको इजी हो, ठीक है। अच्छा, total in our embryology module we will cover these topics. So first will be our fertilization. Okay, fertilization we will study today. Then there are the stages of development. Okay, how embryo will develop, and then what are the structures developing from the germ layers. You all have known the 3 germ layers: ectoderm, mesoderm, and endoderm. This is a very important topic. You cannot skip these structures developing from germ layers. Okay, you may get 2 questions in every exam. You must get 2 questions from 1 or 2 questions. Okay, you have to remember these. Then there is a placental structure. Few questions may come. Then the urogenital system, because it is related to or module as well. So urogenital is very important for this embryology. Then GIT is also very important because there are anomalies like anomalies and the physiological few things, the rotation, malrotation of gut and physiological hernia. So that's also very important. Fallocele, gastrulation, then the cardiovascular system as well, the fetal and fetal circulation as well. And also this is a very important, and it's a pet question for exam, pharyngeal arches. Okay, you will see in this embryology module there are this pharyngeal arches, which are 6, and what structure developed from which pharyngeal arch. So these 2 things you have to remember by heart: the germ layers, the pharyngeal arches. Okay, and you have to memorize these as well. Okay, you cannot skip them. So these 2 are very important. Rest of the things are also important, but there are few things which are important which you have to memorize. Okay, let's start today's topic. Okay, so starting with the gametogenesis, because we are reading the fertilization today. So gametogenesis, the name indicates it is the formation of gametes. Okay, genesis means production. So it is a process in which female and male germ cells, they undergo cytoplasmic and chromosomal changes to form the definitive, what is the germ cell of female? Oocyte and spermatozoon. Okay, so these are the 2 germ cells in the female oocyte, in male spermatozoon. Okay, so this whole procedure of formation of oocyte and spermatozoon is called gametogenesis. Now in females it is called oogenesis, and in male it is called spermatogenesis. Now there are the few differences between male and female gametogenesis. Now you all have known that oogenesis, it is a cyclical event. The monthly female cycle, it consists of maturation of a single oocyte and is accompanied by changes in the hormone secretion. There are concurrent changes in endometrium to prepare the uterus for pregnancy. And when oogenesis it occurs, it only occurs, this oocyte formation is in utero, you can say. You all have known that all the oocytes which have to be formed in the female fetus, they are developed in utero. In females, adult females, there are no oocytes, new oocytes are formed. They are just the oocyte pool is already there in the female ovary. Okay, so oogenesis is only occurs in utero. Oogenesis. In contrast to the spermatogenesis. Spermatogenesis, it begins at puberty and it continues throughout life. Okay, So this is a very important difference between these two processes, oogenesis and spermatogenesis. We in female, we have a fixed pool. You all have known. We will discuss in our urogenital system in detail. The fixed pool. We all have known that the maximum amount of oocytes we have at 20 week, which would be approximately 5 to 6 million oocytes. Okay, and then at birth it decreases to almost a million. And we all know that approximately only 400 oocytes they are to enter the cycle in the reproductive cycles, you can say. Okay, so only 400 basically oocytes are used in a female reproductive age, but we have millions of oocytes in our ovaries. Okay, so spermatogenesis begins at puberty, continues throughout life. Now, both oogenesis and spermatogenesis, they require two meiotic cell divisions, which reduce the number of chromosomes from diploid to haploid. This we will discuss in our genetics module. I have given a lecture on genetics. If anybody wants to study genetics before our module, then you can see the lecture of genetics. Okay, so we will discuss the details of these meiosis and mitosis in that lecture. So all of you have known that oogenesis, spermatogenesis, we have two meiosis. Okay, meiosis 1 and meiosis 2, 1 and 2. Now, what happened during this meiosis? The number of chromosomes are reduced from diploid to haploid. Now, also, what is the next thing meiosis do? It allows the random assortment of maternal and paternal chromosomes and the redistribution of genetic information through the process of crossing over. Now, this is very important because the genes, they cross over from male to female, maternal and paternal chromosomes basically cross over, so that the new genes, redistribution of genes occur in the offspring. Okay, so now this is the fertilization. You all have known, okay, what is the fertilization. So it is a series of events that begins with penetration of sperm into an oocyte and ends with the combination of maternal and paternal chromosomes. Basically, it is a combination of female and male gamete, sperm and oocyte. And then their chromosomes are also combined. Now, where it occurs fertilization, you all have known that it occurs at the ampulla of the fallopian tube. Okay. Now, that's the significance that when we have ectopic pregnancy, the most common area of ectopic pregnancy in tube is ampulla because fertilization occurs here. Okay. Now, that's the next very important point you have to remember, at what time fertilization occurs after ovulation. When the oocyte has ovulated, then when it will occur the fertilization. So fertilization, it will take approximately 12 to 24 hours after ovulation. Okay, so this is important to remember. Now these are the steps of fertilization, that how the sperm it gets to enter the oocyte. Okay, so there are the few changes, few things sperm has to do. Okay, so what sperm has to do? Sperm activation and penetration of corona radiata. Now what happens? Sperm must undergo a process of capacitation, which makes them capable of penetrating the ovum. Now, what is capacitation? In this phase, glycoprotein and cholesterol of acrosomal membrane, they are removed by the secretions from the cervix. Now, when the sperm enters in the female genital tract, now there the capacitation occurs. Okay, so acrosomal membrane, the glycoprotein and cholesterol are removed. Okay, then sperm then undergo an acrosomal reaction, which release the enzyme hyaluronidase, which is needed to penetrate the corona radiata. Okay, so first is sperm activation. It occurs in the cervix and uterine tube. Second is the penetration of corona radiata. For that, it needs hyaluronidase enzyme. Okay, now what will be next? If you see, this is the structure of sperm. This is whole is the head. It is the head, then the tail. So head and tail. So acrosome is basically the anterior most, you can say the topmost part of the head, and then the nucleus, then the mitochondria is in tail, and then this flagella, or you can say tail. Okay, so first would be the acrosomal reaction, the capacitation and acrosomal reaction. Now next, what happens, it is attached to and penetration of zona pellucida. Now sperm binds to the zona pellucida and it releases its acrosomal enzymes. Now for penetration, these enzymes has to be released from the sperm. It is esterase, neuraminidase, and acrosin. Okay, so spermatozoid binds to the zona pellucida and then initiates a zona reaction, which change its physical properties to prevent attachment of any more sperm. Okay, so that sperm which do all these things, that capacitation, the acrosomal reaction, the acrosomal enzymes, then when the sperm it binds to zona pellucida, then this process is called zona reaction. And zona reaction, it prevents further attachment of more sperms. Okay, so that no more sperm can fertilize this egg. Only this first sperm which enters the zona pellucida, only it can go inside to the oocyte. Okay, so this is the diagram. You can say, if you see this diagram now, step by step, they have written sperm makes contact with egg. Okay, sperm, this is the acrosome, the topmost. This is the zona pellucida. Okay, so sperm first makes contact, the acrosome reacts with zona pellucida. You see the hydrolytic enzymes are released. Acrosome reacts with perivital space, then it enters. See, these are the protein receptors. Then what happened? Then the plasma membrane of sperm and eggs, they are fused, which will be discussed in the later. Now, this is the next step, that the fusion of oocyte and sperm cell membranes. Okay, when the sperm attaches to the zona pellucida, okay, after that both the cell membranes, they will be fused. Sperm now comes in contact with the cell membrane of oocyte, resulting in the fusion of membranes of sperm head and oocyte. Okay, so the cell membranes of sperm and egg break down at the area of their contact. If you see this diagram, see, plasma membranes of sperm and egg, they fuse like this. Okay, then it will enter this sperm nucleus. See, the sperm nucleus will going to enter into this oocyte. Okay, now when sperm enters, what happened? The completion of meiosis in the oocyte and formation of pronuclei. We all have known two meiosis phases, and the meiosis in the oocyte, it will complete after fertilization. So soon after entry of spermatozoa, the oocyte resumes second meiotic division, forming a mature oocyte and a second polar body. The chromosomes of oocyte and sperm condense and enlarge to form pronuclei. So basically, this is the at fertilization, the oocyte, in the oocyte phase, it was suspended in a prophase phase. So resumption of second meiotic division of oocyte, it happens after the sperm gain entry into the oocyte, and then the formation of pronuclei. Now, if you see this sperm, it enters into this, and then the sperm and sperm nuclei, and this is the post ovum, they fuse and the fusing nuclei. Okay, so the two cells form the zygote. Now, when the two cells, the sperm and ova, they combine, then they form the zygote. You can say the fertilization is completed. When the male and female pronuclei meet, their membranes break down and chromosomes mix to produce a single cell called a zygote. Now, at this stage, the process of fertilization is completed and the zygote prepares for its first meiotic division. Now, the fertilization is complete, and then the zygote, the cell which make is the zygote, and the zygote now has to undergo multiple mitotic divisions. Okay. Now, this is an important question as well. The first mitotic division is complete at 30 hours post fertilization. Now, we have read up till now two timelines. Like we have discussed that after 12 to 24 hours after ovulation, fertilization occurs. Okay, fertilization occurs after 12 to 24 hours. Now, After fertilization, after 30 hours of fertilization, first mitotic division occurs. Okay, so these two timelines you have to remember. Okay, so first mitotic division is completed 30 hours post fertilization. Now it means the zygote has formed. Now it will undergo mitosis and it will undergo the stages of development. Now these are the timelines at which stage what it would be called. Now from the conception to 2 weeks, we can say it is a pre-embryonic period. In this period, the fertilized ovum it undergoes mitosis, the formulation of morula, blastocyst, it implants, and the germ layers develop. So this 2 week to 8 week, it is called embryonic period. And here the germlines and placenta develop, all the main body systems are formed. And this is the period of, you can say, organogenesis. Because all the systems, all the organs, main body systems have to be formed during this embryonic period. So it is a very, very important period. Okay. Now, from week 9 to birth, it is a fetal period. So you can say that up till 8 week, it will be called embryo. And after 8 week, we can call it as a fetus. Okay. So embryonic period up till 8 week, and after 8 week is a fetal period. Now in fetal period, just the growth and development of organs. So locomotor system, it becomes functional. But the main body organ, body systems must have developed by the embryonic period. Now what happens in the first week of development? Now this is the development stages we are going to read. Now what happened? We have discussed that a zygote has formed. Okay. Now what happened in zygote? There have to be a series of rapid mitotic cell divisions in which the zygote divides over a period of about 3 days, resulting in a so-called 16 cell stage embryo. Now each cell, see, in 3 days it goes to 16 cell stage. Each of this cell is called blastomere. And the solid sphere of cells, like this is the solid sphere of cells, it is known as morula. Okay, now this whole sphere of cells, it is called morula. And if a single cell is called blastomere. Now each of these new daughter cells at this stage is pluripotential. Okay, so it is pluripotent. It can make any system. So every cell is pluripotential. So cavities appear within the center of sphere of cells, forming a blastocyst. Now, first was the morula. Okay. Now, when the cavity forms cyst, the name indicates cyst means a cavity, a cystic structure, a cavity. So when the cavity appear within this sphere of cells, it could be called then blastocyst. Now, see, this is a blastocyst. See the arrangement of cells now. Previously, there were only cells everywhere. Okay, that was the morula. Okay, now what happened in blastocyst? Basically, how the cyst forms, that few cells, they line the border of the cells. Okay, so they form an outer layer of cells and they form an inner layer, you can say. So some cells are combined to form an inner layer. This is the inner cell mass. And then the outer layer of cells formed, you can say that as a trophoblast. And in between this is a cavity appears, which is cavity is called blastocoele. See this? The outer layer of blastocyst soon thins to single cell thickness to become the trophoblast. So this is the trophoblast, the outer layer of cell. Includes the large fluid-filled blastocyst cavity. So this is the cavity, which is called blastocoele. And the central group of cells, this is the inner cell mass. It moves to one pole of blastocyst, it will be called the embryonic pole. Now you can say the major cells, they have gone to one pole. So this will be called the embryonic pole to form the inner cell mass from which the whole embryo itself will form. Okay. Now it has a fluid-filled cavity which would be known as blastocoele, and the trophoblast contributes to the fetal component of placenta. Okay, so this is called the blastocyst. Now, if you see this diagram again, early stages of implantation. Now, what is this morula? Morula is at day 3. Now, we discussed that morula is the whole sphere of cells. Okay, so all the cells are present. And what happened in blastocyst? Blastocyst forms at day 5. Now, blastocyst means a cavity has appeared. Just you remember. So, a cavity appears and there is the outer layer of cell and there is an inner layer of cells. Okay. And what next? Now, next is that blastocyst at day 6, it makes contact with the uterine wall. Now, this was the whole blastocyst. It contacts with this uterine wall. See, this is the uterine epithelium. Okay. So it makes contact with the uterine wall, not implanted, but just contact the uterine wall. Okay, so these are the days you have to remember for exam purpose. They can ask you at which day morula develops. So morula develops at day 3. 3 to 4, you can say just 3. So at day 3, morula of 12 to 16 cells will have reached the junction of uterine tube and uterus. Okay. And day 4 to 5, the fully formed blastocyst, it reaches the uterine lumen. Okay. So coming from the tube, in the tube, it will be in the stage of morula. And then day 4 to 5, it enters the uterine cavity. Okay. They can ask you like this as well. So these 3 days are very important. And implantation just start at day 6. And when it's completed, we'll discuss in next slide. So this was the whole first week of development. So in first week, these 3 things are very important. When morula form, when the blastocyst form, and the implantation just starts. So it just touches the endometrium. Now the second week of development. So the very important thing, what happens during second week? Implantation. Okay, or you have to know the meaning of implantation. That this blastocyst, it has to be implanted into the uterine wall. So implantation and the formation of bilaminar embryonic disc. Bilaminar, the name indicates bi means 2, laminar means layer. So 2-layer embryonic disc would be formed. So this is the whole story of second week. Now starting from day 8, you can say the implantation process, it initiates the decidual reaction or decidualization in the uterine stroma, the cells of which contribute the maternal component of placenta. Okay, so there would be the 2 components of placenta. One would be the fetal component, the other would be the maternal component. Okay, so now what happened? The trophoblast, they begins to differentiate. If you see the diagram, this was the trophoblastic layer we have discussed previously, and this was the cavity. So this outer was came to the cytotrophoblast. Now these trophoblasts now going to differentiate themselves. Okay, inner part and outer layer. Now inner part becomes a single layer of cells, which is the cytotrophoblast, and the outer layer, it would be the, I think I have written it wrong. The outer layer, this should be inner layer actually. It is more extensive and invasive layer. It is called the syncytiotrophoblast. Okay, so it differentiates into 2 layers. One is the syncytiotrophoblast. Syncytiotrophoblast actually has to invade into the, see this layer, it invades into the uterine wall. Okay, and this is the outer will be the cytotrophoblast. And if you see, this was the layer of embryonic embryonic disc or embryonic cell mass. It differentiates into 2 layers. If you see the diagram, it has now in 2-layer form. Upper is the epiblast and lower is the hypoblast. So epi means above, hypo means below. So the name indicates the bilaminar disc appears. Okay, so just implantation has started and the bilaminar embryonic disc has formed. Now, if you see closer, The bilaminar disc, it is derived from the inner cell mass, which is called the embryoblast, and is composed of two layers, the epiblast and the hypoblast. It will be the dorsal germinal layer, it will be the ventral germinal layer. And now, that is a very, very important in this day 8. What happens in day 8? The amniotic cavity, it develops within the epiblast. If you see the previous diagram, see this previous diagram, this was the epiblast. This is the epiblast, is just the layer of cell, and underneath just the hypoblast. Okay. Now what happened when the amniotic cavity appears? Now here, this is the bilaminar germ disc, and amniotic cavity develops within the epiblast. So upward, you can say here, this is the amniotic cavity which develops, okay, within the epiblast, you can say. Now in day 9, what happened in day 9? Lacunar stage begins. Now lacunar stage, you can say, if you see these things, these are lacuna, okay, cleft-like openings. When they fuse, they will form the, hello, okay. So lacunar stage, when they fuse, they will be forming the syncytial trophoblast. It would be invade into the uterine epithelium. Okay, so now what occurs in this day 9? This hypoblast, it forms the exocoelomic membrane that lines the yolk sac. If you see, this is the membrane. If you remember it like this, that epiblast was the upper layer and it forms an upward cavity. That was the amniotic cavity. Now, if you say this below is the hypoblast, it will be lining the lower cavity. This cavity is formed called the yolk sac, and these are the cells. What would be they called? This is called exocoelomic membrane. So this hypoblast, it forms this membrane, okay, that lines the yolk sac. So these are the hypoblast cells which are lining this yolk sac. And now the syncytial trophoblasts, they are invading the uterine glands and capillaries, okay. So these are the uterine glands. So the trophoblasts, they have to enter into the glands. So that was about the day 9. Now day 12. Okay. Now day 12 is very important. The formation of lacuna in the syncytial trophoblast, which we discussed previously, they communicate with the endometrial sinusoids. Okay, if you see these, the trophoblastic lacuna, see they are also combining itself and they form the lacuna, and then they communicate with the endometrial sinusoid. So, and the endometrial blood vessels, they actually invade into them, and then basically the pathway of the uteroplacental blood exchange would be formed. Okay. So for the development of cytotrophoblast, which is thickest at the embryonic pole of the conceptus, now the extraembryonic mesoderm. Now extraembryonic mesoderm, which is derived from the yolk sac cell, it forms, and the extraembryonic coelom forms. Now this is the new thing which happens in the day 12. If you see this diagram, that these are the, you can say, some cavities, the cleft-like openings, they appear, and then they fuse, it would be called the coelomic cavity. So this is the extraembryonic coelom, and it is also known as chorionic cavity. So it was formed by lacuna, which appear within the extraembryonic mesoderm that merge. So these are the lacuna. When they merge, when all these lacuna, they merge, they will form the chorionic cavity. Okay. So outside this whole would be the chorionic cavity, you can say. Okay. So the day 12 was significant with the formation of lacuna. This was important, and the chorionic cavity. These two things are important for day 12. Now day 13, you can say all the implantation process has been completed. If you see this diagram previously, see this was the uterine epithelium. So the whole conceptus has been engulfed by the uterus. So you can say that at day 12 or day 13, the implantation is completed. Now, what happened next? The cytoplasm, they will be forming the primary chorionic villi. the yolk sac, it is now called the secondary yolk sac because a chorionic cavity has been formed outside. Okay, so now the yolk sac would be called the secondary yolk sac, and there's a connecting stalk which connects embryo to cytoplasm. Basically, this connecting sac is the future umbilical cord, and it is derived from extraembryonic mesoderm. Now, the cytoplast basically it forms these villi. You must have known this. And in situ trophoblast, it will be producing the HCG. So you can say at day 13 or at the end of second week. See, the second week is completing now. So what happened is in situ trophoblast, this is release of HCG, human chorionic gonadotrophin, which maintains the corpus luteum in the ovary. So you can say that at the end of second week of development, the HCG concentration increases in blood and the pregnancy can be diagnosed in urine or blood as well. Okay, so pregnancy can be diagnosed at the end of second week. So this was the end of second week. Now the third week of development. Now in the third week, if you remember this name, the gastrulation, it occurs. Now this means that it is a formation of three germ layers. Okay, so this is a very important step in the third week of development. We have discussed previously that we just have two layers. One was epiblast and the other was hypoblast. Okay, or you can say epiblast actually it makes the ectoderm and hypoblast makes the endoderm. Now, where the third layer comes? So basically, this ectoderm it gives rise to intra-embryonic mesoderm. Okay, so the mesoderm is formed from the ectoderm. Now, these three layers they are formed. This three layers, the formation of three layers is called the gastrulation. Okay, so basically, you all have known the ectoderm. It makes the epidermis, the nervous system. Mesoderm means the skeletal tissue, connective tissue, the muscle tissues of the body will be formed. Endometrium, it would be the making the lining of GIT, the urinary tract. Okay, we will discuss in detail all the formation, all the structures which form from these layers. Now, what happens at day 15 means the start of third week. A primitive streak, it appears at the caudal end, and a primitive node appears at the cephalic end. If you see like this is an embryo, okay? So this is a streak, you can say primitive node would be at the cephalic end. So this is the cephalic end. The intraembryonic mesoderm, it lies between the ectoderm and endoderm, except in two locations where ectoderm meets the endoderm, okay? So at these 2 points, you can say that there is no mesoderm, only the ectoderm and endoderm. So upward, you can say cephalic is the buccopharyngeal membrane and the caudal would be the cloacal plate. So procaudal plate and cloacal plate, the intraembryonic mesoderm is not present, just the ectoderm and the endoderm. Now these two membranes, they have to break these membrane. Buccopharyngeal membrane, it breaks down at 4th week, and the cloacal membrane, it breaks at the 7th week. Okay, so okay. So basically the cells from primitive node, they migrate cranially towards the procaudal plate, and this form the notocaudal plate, which becomes a future notochord. Okay, so detail of these embryology, but for that you just need to know need to understand the basic how this is going to develop. Okay, so you can just read these things, that the notochord plate is formed and the notochord will be formed from this. Now the notochord, it comes to underlie the future. Okay, so neural tube will be forming the future brain and spinal cord, and underneath this neural tube would be the notochord. So the notochord, it forms a longitudinal axis of the embryo and the centers of the intervertebral. Okay, so this was the notochord, and then the neurulation. So the name indicates neurulation means that the process of formation of brain and spinal cord, it is called as neurulation. So neural plate is formed at day 18. And caudal ends of the tube are open. They're called the neural. And at the edges of the neural tube, there would be the neuroectoderm, which is continuous with the surface ectoderm, and the neural crest cells are formed. So like this, that you can say this. So these are the neural fold. They just deepens into this like this, and then the neural tube. So and here the edges of the neural crest cells here. So basically these neural crest cells, they then detach themselves from the rest of neural the tube forms, and they form discrete aggregations of neural crest cells. Now the neural crest cells, they also develop few structures. So the brain and spinal cord would be formed from the neural tube. Okay, so the neural tube which is forming these brain and things. Okay, so now the mesoderm. We have actually three, you can say the three types of mesoderm. Paraxial, axial, which is near to the axis, you can say. So this is the paraxial mesoderm. Then the lateral plate, you can say, so lateral mesoderm, and the intermediate. You can say the development of mesoderm, it is in three parts. So paraxial would be middle, midline, then the intermediate, and then the lateral plate mesoderm. Now, how the paraxial mesoderm that develops? The paraxial mesoderm, they undergo differentiation in paired blocks. Like this was the axis, and this paraxial mesoderm, they differentiate in paired blocks like this, you can say. Caudal direction on each side of notochord, it mites. The paraxial mesoderm, it develops into somites. It is a very, very important exam question. Layer the somites form, and when the first pair of somite forms, so at 20 day, the first pair of somites form, and after that 3, you can say, per day they form, and until 40 pairs are formed. So by counting the somites, we can tell the age of embryo as well. Okay, so this is important. You have to know at which day the first pair of somites and how many total pairs of somites form these two things. Dermatomes, you can say, or dermomyotomes, are sclerotomes. So the dermatome, you know, the dermatome and myotomes, which form the connective tissue, skeletal muscles. Sclerotomes, they form bone and cartilage. Okay, so you all have known the mesoderm, development of mesoderm. So from the par, you can say from the par axial mesoderm, what makes the dermatomes, myotomes things, the sclerotomes things, the bone, cartilage, connective tissue, skeletal tissue, all these things are developed from the paraxial mesoderm. Okay, so now the lateral plate mesoderm. Now we told you that it is continuous with extraembryonic mesoderm. It gives rise to two layers. One is the somatic mesoderm, splanchnic. So one mesoderm, it lines the amnion, and the other lines the yolk sac. We discussed previously that upward was the amniotic cavity, below was the yolk sac. So you can remember like this, that one is the one lining is the somatic and other is the splanchnic. So the two layers, they enclose the intraembryonic coelom, and it is the forerunner of the serous cavities. Okay, so all the pericardial, pleural, peritoneal cavities, they develop in the intraembryonic coelom. Okay, so the intermediate mesoderm, it just forms the urogenital system. So all our urogenital, the urinary tract, the kidneys, the genital organs, everything will be developing from the intermediate mesoderm. Now, this is a very important. I have written down all the structures which are developing from ectoderm. So you all have known the structures from the ectoderm. See, every skin, feature, you can say epidermis, hair, nails, lens, cornea, sebaceous glands, tooth enamel, epithelium of mouth and nose. Okay, so all of you must have known that, okay, this is formed from the ectoderm. Okay, so this is also ectoderm from neural crest cells of ectoderm. We discussed neural crest cells. Where were the neural crest cells? It was just the neural lateral to the neural tube. Okay, so neural crest cells, what develop from the neural crest cells? They develop into, the name indicates, neural things, something. So all the nerves, all the peripheral nervous system will be developing from these neural crest cells. The adrenal medulla, it is also nervous. The melanocytes, the facial cartilage. These two things are different: the facial cartilage and the dentin of teeth. Okay, so these are also forming from the neural crest cells. And then the neural tube. So neural tube, we discussed that it is a part of ectoderm. So ectoderm is developing into the skin, the neural tube. So neural tube is forming all the neural structures, the brain, spinal cord, now posterior pituitary. We'll discuss in endocrinology as well. The posterior pituitary is actually the evagination of, you can say, the hypothalamus, you can say. So the motor neurons, the retina, the anterior pituitary. So all these things, the neural tube is also developing from ectoderm. So these are the structures from the neural tube. Now the mesoderm. Now from mesoderm, all the muscles, the bone, cartilage, connective tissue, adipose tissue, lymphatic system, dermis, genitourinary system, and serous membranes. Okay, so everything, you can say, in the middle, the muscle, bone, cartilage, like things, they are forming from the mesoderm. Now I have told you three types of mesoderm. One is the intermediate, the paraxial, and the lateral plate mesoderm. So the intermediate mesoderm, you just need to know that the urogenital system. So the uro, kidney, and the gonads, it's the genital. So only the urogenital system is developing from the intermediate mesoderm. This is a very important exam question as well. You have to remember this. And also this, the paraxial mesoderm. So then paraxial mesoderm, we discussed that dermatome, sclerotome, myotome. So the name indicates cartilage, skeletal muscle, connective tissue, dermis. So these type of things are developing from paraxial mesoderm. And then the lateral plate mesoderm. So through the lateral plate mesoderm, the circulatory system, the spleen, the gut wall, they form from the lateral plate mesoderm. Okay. Now the endoderm. So endoderm is actually very, very easy. So all the inner linings, you can say the stomach, the intestine, mucosal layers, colon, liver, pancreas, bladder, epithelial parts of trachea, lungs, pharynx, thyroid, parathyroid. So basically all the inner organs, you can say, the GIT organs, the liver, pancreas, they develop from the endoderm. So these slides are very, very important. Mesoderm, the structure developing from ectoderm, mesoderm, and the endoderm. Now you have to remember these three slides by heart. They can ask you anything from these slides. Okay? Now coming to the next week of development, we have discussed third week of development. Now coming to the fourth week of development. So fourth week of development would be the very important thing. Fourth week, it would be the folding of embryo. So basically, embryo folds in two planes. One is the longitudinal and longitudinal, like this folding, and the lateral would be the transverse folding, you can say. So lateral folding, why it happens? It happens due to the enlargement of somites. So basically, the paraxial somites, they actually enlarge. So that's why these would be the fold. These fold inside. Okay, so that would be the lateral folding. We don't need to go into details of these folding. So this is the longitudinal folding. In the longitudinal folding, this is the head end. You can say this is the tail end. Both these fold on itself. So basically, longitudinal folding occurs mainly as a consequence of rapid enlargement of cranial end of neural tube to form the brain. So the cranial end, it enlarges. So that's why the rapid enlargement of foramen. Also, that's why there is a longitudinal folding. It occurs between day 21 and 24. So the embryo is expanding and the head and tail are brought closer together. And if you see this, that it was the yolk sac, you can say, is connecting to the midgut. So amniotic cavity pinches the connection of the yolk sac and gut to form the narrowed communication of vitreointestinal duct. So these just narrow down and they just narrow down, and then only the vitreointestinal duct it is formed, and later also this duct is lost also. Okay, so yolk sac would be separated from these things. Okay, so this was all about the development of embryo, you can say. And now we are going to discuss the development of placenta. So basically, placenta has both maternal and fetal components. So you can say maternal parts are derived from decidua basalis. So basalis means the basal layer of endometrium. So decidua basalis and fetal parts, they consist of villi of chorion fundosum. So basically the villi, you can say. So villi forming the fetal parts of placenta and maternal parts forming from the decidua basalis. And you can say the placenta has two units. One would be the chorionic plate and one would be the basal plate. So basal thing would be on the maternal side, chorionic plate would be on the fetal side, you can say. Okay. If you see this diagram, this is the umbilical cord it inserted. And what happens, what is this? This is the chorionic plate. This would be the chorionic plate, the inner layer which is facing the embryo. And if you say this would be the basal layer, basal plate. So this would be the placenta layer would be the basal plate, which is facing the maternal side would basal plate, facing the fetal side would be chorionic plate. Now the chorionic plate, it is of fetal origin and is composed of you say amnion, extraembryonic mesenchyme, cytotrophoblast, and cytotrophoblast. Okay, so all the trophoblasts, they are going inside, so making the chorionic plate, and it is of fetal origin. So the basal plate of now the basal plate, it is made of fetal derived parts and maternal derived parts. So basal plate, you cannot say the basal plate is just the maternal in origin. The basal plate also has two parts. One would be the fetal parts, other would be the maternal derived parts. So fetal parts would be the cyto and syncytiotrophoblasts. Okay, because the villi they go inside, so the cyto and syncytiotrophoblasts are present. And then the maternal derived part would be from the decidua basalis. Okay. So now the villi. So now the villi are their chorionic projections suspended in the intervillous space. You all have known that purpose of villi is to maximize the area of interchange with the maternal blood. Each contains a capillary plexus supplied by branches of umbilical vessels. Interchange occurs with maternal blood brought to the intervillous space by branches of uterine vessels. If you see this diagram, that this is how the umbilical cord. Okay. So this is how the umbilical vessels they enter. This is the umbilical artery and vein. They enter the chorionic plate, and see, if you see, this is a tree. This is basically a chorionic villus tree, you can say. So these villi they just give branches, multiple branches, and then they open into the intervillous space. So maternal blood supply is in the, this is the intervillous space. So maternal blood supply and fetal blood supply, you can say the exchange of materials, they occur in the intervillous space. Okay, so how the chorionic villi they develop. Now coming to again the day 12. So day 12 would be the second week. We discuss the formation of primary chorionic villi. Now the primary stem villus. Again, there would be multiple types. See, this is the chorionic plate. Okay, chorionic plate, the villi starts. First is the primary stem villi, then it give branch, it will be forming the secondary stem villi, and then the tertiary stem villus. And if you see this tree, they are the placental villi. These are the free villi, you can say so. And these villi, they open into the intervillous space. Okay. So, how the villi form? The clefts appear in the syncytiotrophoblasts called lectina. which communicate with maternal endometrial sinusoid. So cytotrophoblast forms primary chorionic villi. Okay, so cytotrophoblast, they forms this primary chorionic villi. Now at day 15, if you can remember it like this, at day 12, primary villi, day 15, secondary villi. Now, how the secondary villi form? The extra-embryonic mesoderm, they invades the core of primary villi, converting them into secondary villi. Okay, so in secondary villi, one thing added on, the extra-embryonic mesoderm it adds. So secondary villi outline the entire chorion. Now, tertiary villi, day 21. So 12, 15, and now 21. Primary, secondary, tertiary. So tertiary villi form. Now, what has additional feature in tertiary villi? The villi will contain the blood vessels. Okay. Now 21 and then 28, you can say free villi form. Cytoplast in the tertiary villi disappears. Chorion leaf is formed from chorionic villi located on the embryonic pole. These become compressed against the decidua and become avascular, and chorion frondosum is located at the embryonic pole. Now, this would be important here. So the tertiary villi of chorion frondosum, it grows towards the basal plate and attached to the decidual tissue via the cytoplastic shell. Now, if you see the tertiary villi, it actually grows toward the basal plate and it actually attaches to the decidual tissue. Okay. So if it attaches, then it would be called as the anchoring villi. See this anchoring villi. So if you see this diagram, what color is it? Yellow. So this was the basic stem cell. Stem cell was the primary villi, go to secondary, tertiary, pre-villi, and then the villi, they attaches to the basal plate. See, this was the basal plate. This is the basal plate. So this villi, they attach to this basal plate to be called the anchoring villus. So the anchoring villi, they give rise to side branches called intermediate villi, which in turn produces the terminal villi. So the terminal villi, they develop as sprouts of syncytial trophoblast. They take on function from intermediate villi in third trimester of pregnancy. Now the intervillous space. All this is the intervillous space. If you see this, this space is called the intervillous space, where the villi are opening into. So it's a blood-filled space between anchoring villi, also known as trophoblastic lacunae. If you see, this is the basal plate, the maternal artery. See, this is the artery and vein. This would be the maternal artery and vein. Okay, so uterine artery, maternal uterine arteries are here. So uterine artery, basically they opens into this intervillous space. So here the exchange of blood and all the materials would occur, because this anchoring villi, they also open into this intervillous space. So this is the blood-filled space between the anchoring villi. Okay, now this is the placental membrane, you can say placental barrier. Now what happened, it is the partition between the fetal and maternal circulation. Now how it partitions? So if you see the diagram, see, this is the embryonic blood vessels. So these are the blood vessels, branches of the umbilical arteries. Okay, so it composed of fetal tissue. It initially has 4 layers. Now, what would be the 4 layers? So the blood supply of fetal, you say, first would be the fetal capillary endothelium. If you see the diagram, first would be this basement membrane, you can say, or the capillary endothelium. Then would be the connective tissue of villi. See, this is the connective tissue. The second layer would be connective tissue. Then would be the cytotrophoblast. So third, this is the cytotrophoblast. Then would be the syncytiotrophoblast. The fourth one layer is the syncytiotrophoblast. And outside this is the maternal blood is surrounding the chorionic villi. So maternal blood, basically it opens into the sinuses, or you can say the intervillous space. So these are the placental membrane, you can say. So the fetal blood is inside the fetal capillaries. So the exchange occurs through this, the fetal capillary endothelium, then the connective tissue, then the cytotrophoblast, syncytiotropoblast, and then the maternal blood. Okay. After the 4 months of fetal life, placental membrane, it becomes thinner due to villus maturation. So it becomes thinner later on in the pregnancy. If you see this diagram, so again, it has discussed that this is the capillaries. So the basement membrane or capillary endothelium, then the connective tissue, then the syncytiocyto, both layers you can say, and then the maternal blood sinus. So this is a placental barrier, the barrier through which the exchange of materials occur from the fetus to the mother, or from the mother to the fetus. Okay. So this is umbilical cord. It is very important. So what is the total length of umbilical cord? It is approximately 50 to 60 centimeter with diameter of 1.5 to 2 centimeter. And it contains, what it contains, we have 2 umbilical arteries and 1 umbilical vein. Okay. And the Wharton's jelly, it's just the, you can say, a jelly-like material which is between the 2 arteries and vein. Okay. So 1 in 100 newborns, 1% you can say only have one umbilical artery, then 20% of them will have a coexisting vessel or heart abnormality. Okay. So it usually, basically umbilical cord, it normally inserts in the placenta near the center. If you see this diagram, this is the central attachment of umbilical cord. See, umbilical cord attaches here. Here in eccentric, umbilical cord is just off center. Okay, so just off to the center. And this is markedly eccentric as well, and marginal. If marginal means that just near to the margin, okay, within 2 centimeter, you can say it is attaching. So if you read this, the eccentric means the cord insertion is off center, 2 centimeter away from the edge. Marginal means within 2 centimeter of the edge of placenta. And filamentous, it is very important. Filamentous mean cord inserts into the membranes and then travels within the membranes to the placenta. So the exposed vessels are not protected by Wharton's jelly. So the filamentous cord insertion, it can be making the vasa previa risk factor in vasa previa, that the filamentous cord insertion, the cord is inserted into the membranes. Okay. So in vasa previa, this type of cord insertion happens. Okay. So now we will be discussing the types of twins, the how the twins forms. We have discussed that their how the zygote form. This is the sperm, then this is the egg. Okay, so these two both they form a zygote. Okay, so one sperm, one egg, they fuse and form a zygote. So this is the normal thing. Now one word you have discussed, that dizygotic. So monozygotic or dizygotic, there are 2 types of twins. So monozygotic means the name indicates one zygote, but the dizygotic means 2 zygotes. So basically, one sperm, one egg, it makes a one zygote, and there's another sperm, another egg, which make a another zygote. So basically, the body has in the fertilization process, the 2 zygotes are already formed. Okay, and they both goes on to develop into the fetus. So this would be the twin pregnancy, and you can say the dizygotic pregnancy. So all dizygotic twins, they would be dichorionic. So dichorionic, you must have read the dichorionic, diamniotic, or monochorionic. So dichorionic means, chorionic means, you can easily remember the placenta or the chorionic membrane. So the placenta, you can remember it like this. So when 2 zygotes are formed, it means everything would be different. There would be no similar twins. They would be not similar because they are they are developing from different zygotes, different germ cells, you can say. So the 2 zygotes, when the dizygotic twins are formed, they would be the fraternal twins, you can say, and they would be non-identical twins, and they both have two placentas. They both have separate placentas. Now the monozygote means, it means that only one zygote was formed. Okay. Now how the twins formed from the one zygote? Now this happens that it divides. Now if the division occurs at morula stage. We discussed that morula stage was at day 3. So if at day 3 they divide, they split basically, because they undergo into rapid mitotic division. So if they just separate down, the few cells they go to this side, you can say few cells they go to this side up till day 3, less than 3 day. Okay, so these two cells they would be forming. So the two, you can say now the two zygotes, or you can say the two embryos are developing. So they would be having dichorionic, diamniotic. They would be having like dizygotic. They would be having their separate placenta and separate amniotic cavity. Okay, so two placenta and two amnion would be having. Okay, now what happened when the separation occurs at day 4 to 8? Now after 3 days, you can say day 4 to 8, now it would be called the monochorionic diamniotic. Monochorionic, because the chorion basically it has formed already. So only one chorion would be there and diamniotic. Now the partition would be 2, the amniotic cavity would be 2, but chorion would be single, you can say. Placenta would be 1. So placenta or chorion would be 1 and amnion would be 2. Now, if the separation occurs after amnion is formed, now amnion is formed between day 9 and 12. If the separation formed after the amnion is formed, then only the twins would be having monochorionic, monoamniotic, means the chorion is already formed, the amnion is already formed. So both the embryo will be lying in one amniotic cavity, and they would be having one placenta, or you can say one chorion. Okay, so these days are just important to remember, because these are just the developmental, you can say, process. If the separation occurs before day 3, then each embryo would be having two placenta, di. You can just easily remember dichorionic, diamniotic. If they develop this division or separation occurs between day 4 to 8, means chorion would be 1, MC 1 mono. Okay, amnion would be 2. And if the division occurs between day 9 to 12, then only the single pregnancy, the monochorionic, monoamniotic, one cavity. Okay. And now if they are separated after day 12. Now at the day 12, everything actually completes. So amniotic cavity would be 1, chorionic cavity would be 1. Now after day 12, now what happened? It would be the conjoined twins. So the twins would be, there would be one thing in one twin and the other thing would be having in the other twin. So they would be the conjoined twins when the separation occurs after day 12. So these days are important: 3 day, 4 to 8, 9 to 12, and after 12. Okay, so very easy way to remember. Lesson 3, then 4 to 9, you can say 4 to 8, वैसे तो 9 के साथ ठीक रहेगा, then 9 to 12 and more than 12. Okay, so 3 multiples is, you can remember 3, 9, 9 and 12. Okay, so these days are important. Okay, now this is a very, very important exam question as well. How the twins, the chorionicity, it is diagnosed on ultrasound. So basically chorionicity is better determined by ultrasound before 14 weeks of conception. Okay? So there will be two signs. One is lambda sign, the other one is T sign. If you see the diagram, it is a lambda sign. Lambda signs means the fetus has two chorionic and two amnion. Okay, so the partition line would be thick. If you see this, chorion, amnion, and then here would be the placenta. So this is a lambda-like sign. Okay, you know, lambda sign like this. Okay, so this is the thick membrane between the twins. And what it, so it is the diagnostic for dichorionic diamniotic. And what happens in monochorionic diamniotic? It is just a thin straight line between the amniotic cavities. Okay, like this. This is the two amnions and just a thin line. And you see this diagram, this usually comes in exam. They give you picture and they tell you, okay, what sign is this? So this is a very, very important, very easy picture to clear in the exam. So if you see this picture, this is the lambda sign. It's a very easily, very correctly shown in this figure. See these two fetuses, and there, this is the intersecting membrane. So this is the lambda sign, and this is whole is the placenta. Okay, so basically two placentas are fused actually, and they form this thick membrane between the two ends. And if you see this diagram, this is the T sign. Okay, so this is a very, very thin line between two cavities. So this is the T sign. So T sign, it shows the monochorionic diamniotic. Lambda sign shows dichorionic diamniotic. Okay, monochorionic monoamniotic, what would it be having? It would be having no partition because the both twins, they lie in the same cavity. Okay, so no partition for monochorionic monoamniotic twins. Okay, so this is a very important slide. And then the I think last slide would be of molar pregnancy. Molar pregnancy, it is characterized by presence of hydatidiform mole, and its prevalence in UK is 1 in 700 births. It has two types, complete mole and partial mole. Okay, so complete mole means there is no fetus. Okay, no fetal tissue. It is just characterized by diffuse swelling of villus tissue and diffuse trophoblastic hyperplasia. Diffuse means all the villus tissue are swelled, all the diffuse trophoblastic hyperplasia. In the absence of embryonic or fetal tissue, there would be no fetus or no fetal pole. Okay, only the villus hyperplasia, you can say trophoblastic hyperplasia, would be present. Now, how it happens actually? There are the three possibilities. Okay, now one thing you have to remember, if you see this diagram, empty egg, empty egg, empty egg means that maternal oocyte, or you can say okay, maternal oocyte, it just lost his chromosomes. So no maternal chromosomes are present. It's just the sperm, it enters into an empty egg and then it duplicates. Okay, because only the sperm material is present, genetic material is present, 23X, and then it just duplicate into 46XX. Basically the karyotype would be normal karyotype, you can say 46XX. You all have known that it's a female, normal female karyotype. See again 46XX and see 46XY. So it's a male karyotype. But the problem here is that there is no maternal genetic material is present. So if one sperm it enters into empty egg, it duplicates. There's a possibility that two sperms, they just go into an empty egg and they form this 46XX, or they two sperms with one X and one Y, it goes and forms a 46XY. So it occurs when an empty egg is fertilized by one or two normal spermatozoa. So dispermic fertilization actually, it is less common. This dispermic is in less than 20%. And most common is monospermic. That what we discussed, that it monosperm, one sperm it goes into duplication. So it is most common. It occurs in 80% of complete mole. So they would be diploid. Diploid means 2N status, the ploidy status. So 46 chromosomes. So the karyotyping is normal, just like normal. So genotype is just like normal female, normal male Okay, and all nuclear genes are paternal. You must remember that empty egg, there is no maternal genetic material is present. Okay, so this is about complete mole and the partial mole. Now partial mole means half mole, so there would be a fetus, so fetus would be present here, so that's the very important difference. And then see the word focal swelling. Focal swelling of focal tissue, villus tissue, focal hyperplasia, trophoblastic hyperplasia. In complete mole, we discussed that diffuse swelling, diffuse hyperplasia. In the absence of embryonic tissue, in the complete mole, and in the presence of embryonic tissue, in the partial mole. Okay, so what happens here actually? If you, okay, just read this slide first. Here the egg would be present. So normal haploid egg is fertilized by 2 or 3 spermatozoa. Okay. Here egg is also present and sperms are also multiple, 2 or 3. So the karyotyping or ploidy status, here they are triploid, means 3 sets of chromosomes like 69. So 3 sets means 3n chromosomes. And the genotype would be 60n, 60n carry, 69, sorry. 69, okay. So 69 would be XXY, triple X, or XXY. If you see this diagram, this is the first one is the complete mole. First two are, sorry, these two are complete mole. In complete mole, we have told that no maternal DNA, no maternal DNA. If one sperm goes and they duplicate, so it means the 46XX or 46XY. Now in partial mole, if you see this partial mole, egg also has a 23 set of chromosome, one set of chromosome, one sperm or two sperm, okay? They just add, and one would be the X, and there are two sperms also goes inside. So the formation of maternal and paternal chromosomes, triploid, 3 sets of chromosomes, okay? So 2 or 3 sperms, they enter into one egg. Now the triploidy status is present in partial mole, okay? So that would be the just main differences between complete and partial mole. Now these pictures are also very important for exam purpose as well. They usually come in exam. If you see this picture, you see this is a normal fetus is present, okay? And this is the snowstorm appearance, you can say, in the placenta. So that would be, you can say it is a partial mole, okay? And if you just have this picture, this is the snowstorm appearance here, like this, okay? So this is a very important picture of complete mole. So these two pictures are very important. For example, they can ask you, because in MRCOG part 1, they usually give you pictorial diagrams as well, and they ask you what structure is this. They usually just ask you about labeling of the structural diagnosis as well. So twin lambda and peak signs are very important, and the molar pregnancy ultrasound pictures are very, very important. Any question up till now? We have finished or today's module, today's not module, today's lecture. If you have any question, you can ask, or just go through few questions, okay? No question? Okay. So Dr. Sana, Dr. Sanya, Dr. Nazish, okay, both 3 are present, okay? Now you have to tell me answers to these questions. How many days after fertilization does the blastocyst form? Who will going to answer this? Blastocyst. No answer. Okay, if you see the picture and the very important slide I told you, see, day 4 to 5, the fully formed blastocyst, it reaches the uterine lumen. So that is a very important day. You just have to remember, okay? So you can say that it is a Fifth day, the blastocyst form. Now, morula form. How many days after fertilization morula form? It form 3 4 day, 3 day, you can say. Okay. So, from which germ layer does the pancreas develop? So, all of you must have known that pancreas or all the GI organs, just easy way to remember, they are forming from the endoderm. And this question, when does oocytogenesis is complete? So oocytogenesis means oocytes. So when it would be completed? So it's complete before birth. Before birth, you can say, because it is just this oocytogenesis is just doing in the intrauterine life. It is not going to occur after birth. Okay, now somites are derived from which germ layer? So you all have known the somites are from the mesoderm, and which layer of mesoderm? So this is the paraxial layer of mesoderm. And how many days after fertilization does implantation typically occur? So basically implantation, it completes at actually 12th day. We just discussed that it is in the second week it starts and it completes at the end of second week. Okay, so that's the end of our today's lecture. If you have any question, you can ask. कोई problem किसी चीज में?